DNA is located in every cell and in every cell nucleus. Every cell in your body has the same DNA in it. DNA is in a structure called a double helix, which has two strands that twist around each other. It is made of all the nitrogen base pairs, sugar, and phosphate. The nitrogen base pairs are organized like prongs on a ladder. The four nitrogen base pairs are A, T, G, and C. A pairs with T and G pairs with C. Hydrogen bonds are formed between hydrogen atoms, connect the base pairs. There are roughly 6 billion base pairs in every cell of your body. Deoxyribose sugar is modified sugar that lacks an oxygen atom. The phosphate backbone is a structural aspect of DNA that gives it form, and is made up of 5 deoxyribose sugars and different phosphate groups. 5' and 3' are the carbon numbers in the DNA's sugar backbone. The 5' carbon has a phosphate group attached to it, and the 3' carbon has a hydraulics group. This gives a DNA strand a direction to go in. In order to replicate, DNA enters enzyme complex at a replication fork. Then, helicase separates the two strands by spinning them to unravel them. The 3' strand is the leading strand and is used as a template for replication. The 5' strand is the lagging strand. The leading strand, once helicase separates the DNA, RNA primase creates the opposite base pairs for the first couple of original base pairs so that DNA polymerase can read it and replicate it throughout the entire strand. DNA polymerase then fills in the missing complementary base pairs to create 2 identical DNA strands. The lagging strand goes in the opposite direction, so instead of a steady line, it is replicated in segments. The RNA primase replicates 4 segments at a time, called Okazaki fragments, and then the DNA polymerase fills in the missing segments. DNA ligase puts the replicated strands together.